Prenatal physical development, the 40-week journey from a single-cell zygote to the complex structures and systems that comprise the newborn human involves many steps and processes under the influence of both genetic and environmental forces. There are three main phases in prenatal life: the germinal period, 0 to 2 weeks, the embryonic period, 3 to 8 weeks, and the fetal period, 9 to 40 weeks, see Figure 4.2. The main features of physical development in these periods are summarized in Table 4.1 below. Another mapping of prenatal life is by trimesters, 0 to 3 months, 4 to 6 months, and 7 to 9 months. You can read in more detail about landmark events and features of the physical aspects of prenatal development elsewhere, Moore et al. 2000, Moore and Prasad, 2003. Germinal period, creation of embryonic disc inside the blastocyst, which will form the human being, whilst an outer shell protects it. Embryonic period, heart begins to beat. Not yet a viable organism outside the uterus. Development of neural tube which will eventually form brain and spinal cord. Neuron production begins and neurons migrate to correct parts of body and brain. Development of buds for arms, legs, fingers and toes. Fetal period, body and nervous system begin to operate in an organized way, breathing, swallowing and urinating. Frequent movements of limbs, sex can be determined, may survive premature birth from 21 to 22 weeks. By week 24 most brain neurons are formed. Massive growth. Prenatal development of the brain. Before considering how the brain develops in utero, it is useful to describe some of the main brain features and structures. A quick tour of the brain, some important brain features and landmarks. The human brain consists of billions of tiny cells called neurons that transmit signals to one another in organized networks. They have a typical shape that helps them communicate with each other, looking a lot like tiny trees, comprising a head or cell body, with branches called dendrites, a tail or trunk called the axon and root-like extensions called terminals. When stimulated, neurons propel a tiny charge or impulse, the action potential, from the head to the terminals. This impulse passes from one neuron to another across tiny gaps called synapses, with each neuron potentially having thousands of such connections. Chemicals called neurotransmitters, e.g. serotonin or dopamine, are needed in most cases to help pass the message over the synapse. These connections allow complex networks of neural activity that underlie the functions of the brain and nervous system. See Pinal, 2008, Pervs et al., 2008. The older hindbrain regions are responsible for basic survival functions, states of alertness and well-learned movements, and the midbrain for rapid response to sensory signals. The forebrain has internal structures such as the hippocampus for working memory and aspects of emotional processing, and a thin wrinkled outer layer, the cortex, for most higher-level perceptual, cognitive and emotional analysis and for complex aspects of memory and learning. These areas of the brain are illustrated in Figure 4.3b. The cortex is described in 4 lobes corresponding to the skull bones, as illustrated in Figure 4.3a, frontal, parietal, occipital and temporal, and to some degree, there is specialization of functions in different regions of the cortex, e.g. primary visual processing occurs in the occipital cortex, see Pinal, 2008 or Pervs et al., 2008 for more details. Stages of prenatal brain development. The brain develops very early in utero in stages, Huttenlocher, 1990, Johnson et al., 2002, Nowakowski and Hayes, 2002, Strachan and Red, 2003, Rosenzweig et al., 2005. The basic pattern and timetable is under genetic control. About 40% of the structural or worker genes in the human genome are exclusively involved in brain growth and maintenance, while the other 60% are also involved to some degree as well. Pennington, 2002, Ramus, 2006, but prenatal environment can also impact on this early development of the brain. During the embryonic period, the embryonic disc develops into a three-layered sandwich of cells comprising an outer layer called the ectoderm, a middle layer called the mesoderm, and an inner layer called the endoderm. The ectoderm will eventually form the skin, hair, and nervous system. By day 15, a small group of cells begins to grow rapidly at one end of the ectoderm, creating a primitive basis for the head and brain. By weeks 3 to 4, the development of the nervous system begins in earnest. By day 18, chemical signals are sent to the ectoderm from the layer below, causing the growth of a thick neural plate. The cells in this plate continue to multiply rapidly, although at uneven rates, so that by about day 20, the plate has a groove all along its midline with folds of tissue either side, like a river bed with canyon walls, see figure 4.4. By the start of week 4, about day 22, these folds arch over the groove, wrapping around to form a hose-shaped structure, the neural tube, the precursor to the brain and spinal cord. This folding starts at the middle of the groove, moving towards each end in a zipping manner until the tube is sealed except at the ends. The tube opening at the anterior, head, end now closes, followed by that at the tail end. In about 1 in 1,000 births, Bukatko and Daler, 2004, the neural tube fails to close either at the head end, resulting in anencephaly, a severe impairment of brain development usually fatal after birth, or at the tail end, producing spina bifida, which can result in paralysis. By about 3.5 weeks, there is a defined swelling at the top end of the tube, and by week 4, this shows 3 clear divisions corresponding to the future forebrain, midbrain, and hindbrain. The entire nervous system becomes a clearer structure as the neural tube separates from the surface ectoderm. Many different genes control this process. For example, forebrain development involves genes from the EMX family. After the closure of the tube in week 4, neural development proceeds frantically in several phases which can overlap in different brain areas. The first phase is neurogenesis, the rapid and teeming production of neurons within the neural tube. Neurons cannot copy themselves but are made from other cells in the neural tube that rapidly divide to form more cells, which are in turn changed under genetic control into neurons. Neurogenesis begins in weeks 3 to 4 and is especially active up to week 16, with hundreds of thousands of neurons being made each minute and most of the brain neurons formed by about week 24. This frantic proliferation of neurons is reflected in the corresponding growth in the size of the infant brain, with the head enlarging to 1/2 of the body size by week 8. Neuron production generally ceases at birth, although some new neurons may be made in the adult brain, possibly in the hippocampus, the part of the brain important in making new memories, Erickson et al., 1998. Most brain development in the final months of prenatal life, and after birth, consists of the growth of connections, synapses, between neurons and of the myelin tissue that coats the neural membrane, see below, leading to a tenfold increase in brain weight from 20 weeks to birth. The second important phase is neuronal migration, in which the neurons move to different locations in the emerging brain and nervous system. Some only go short distances but others have to travel far. By a pushing or pulling process under the control of genetic and biochemical factors, they move along fine filaments, sometimes swinging like monkeys from one filament to another. Fong and Walsh, 2001, Hatton, 2002, see figure 4.5 and 4. Movies of migrating neuronal cells, see http://hatton-server.rock-efeller.edu/hattonlab/movies.html. Migration begins in earnest after the final sealing of the ends of the neural tube. This exodus of neurons establishes different neural groups or populations that will eventually contribute to different structures and systems in the brain and nervous system. If this migration is disrupted for any reason, genetic or environmental, then there may be impairment of subsequent brain function. Numerous disorders such as dyslexia and autism have been linked to the impairment of neuronal migration in the prenatal brain. Watts, 2008, see Chapter 16, Atypical Development, for more on autism. The third phase, neuronal differentiation, begins when genes further modify the newly arrived cells to make them specialized for their future roles in the brain regions and systems. During the process of embryonic cell differentiation, two other phases also occur at the same time. One of these is synaptogenesis, the development of potential connections or synapses amongst the emerging neurons due to the growth of extensions or branches at the top of the neuron called dendrites and at the bottom called terminals. There is also the beginning of myelinization or myelination, the coating of the axons in fatty insulating material to improve their efficiency, although this process increases markedly after birth with use of the brain. See the postnatal development of the brain. Pages 88 to 90. In the prenatal brain there is considerable oversupply of synapses. This leads to the final phase or aspect of neural development, involving pruning of connections and rivalry amongst neurons for target neurons. Success in this regard may be due to good availability of connections or even to repeated stimulation of the lucky neural pathways by environmental factors such as sensory input. See next section. This whole process of synapse building and pruning does not only occur in utero, but will also continue vigorously after birth, and to some extent across the lifespan, in response to input from new learning and experience. See the postnatal development of the brain. Pages 88 to 90. The basic brain structures are in place early in the fetal period by weeks 9 to 10, with the main aspect of development thereafter being the growth in brain mass. During the last few months of prenatal life, the cerebral cortex of the brain shows increasing folding due to neuron production and growth of synapses and myelin. Not all regions of the brain develop at the same pace and, even within the cortex, there are different timetables for different regions with these discrepancies continuing after birth. See the postnatal development of the brain, pages 88 to 90. As a result of this rapid development of the brain and nervous system, even the embryo soon displays basic behaviors and responses reflecting organized neural activity. Prenatal states, fetal sleep and waking cycles. By the end of the first trimester, the fetus shows daily, 24-hour or circadian, rhythms of heart and lung activity, and hormone release. The brain center that is critical to circadian rhythms, the suprachiasmatic nuclei, is present by the middle of prenatal life, although circadian rhythms continue to develop after birth. Rivkes and Howe, 2000. Most importantly, sleep-wake cycles have begun. Sleep cycles with rapid eye movements, REM sleep, appear at about 28 weeks gestational age, with REM accounting for most sleep cycles at this time, declining to about 50% by birth. Graven and Brown, 2008. Sleep is not just a passive process. REM sleep is critical to the early development of the brain, especially the organization of the visual, auditory, and touch systems and the ability to form long-term memories. Graven and Brown, 2008. REM sleep deprivation in fetal and early postnatal life is associated with delayed or disrupted development of these capacities. Prenatal abilities and behaviors. One of the most fundamental human capacities is the ability to sense and respond to a stimulus external to the body. Imagine how isolated you would be without your senses to connect you to the world, or how frustrating it would be not to be able to react to some stimulus. But what is it like for the developing fetus? Is there any sign that the fetus can sense and react to the world? Indeed there is, The rapid development of the brain and nervous system supports the early emergence of sensory capacities and behavioral responses, and even within the cloistered world of the uterus, these basic abilities are exercised and refined as the infant develops. By the end of the embryonic period, there is already the capacity to react to external stimulation, initially with simple reflex responses, not engaging the brain, but increasingly with responses under brain control. The 5 main sensory systems of vision, touch, audition, taste and smell all begin to develop in the embryo. All such channels involve bodily receptors, eyes, ears, etc., as well as specialized neural pathways and brain regions. For example, processing in the visual system begins with the components of the eye, lens, retina, etc., continues along the optic tracts to the brain and involves further processing in the visual cortex and other subsequent brain areas, see Figure 4. 6. These systems operate to some extent and provide channels for perceiving even in fetal life, Hepper, 1996. Moreover, as the brain develops across the fetal period, there is an increasing ability to retain this sensory information, which shows the capacity for memory and learning, Hepper and Leader, 1996. Fetal touch, taste and smell perception and memory. The earliest sensory channel to develop is that of touch. During intrauterine diagnostic procedures, it has been established that by 8 or 9 weeks, the fetus moves its head if touched in the mouth region, and by 12 weeks will grasp at anything touching the fingers, Pfeiffer et al., 2001. The fetus regularly receives tactile stimulation via the maternal abdominal wall and will show increased activity if this is touched, Lagerkrantz and Shanjus, 2009. The taste systems and receptors, taste buds, are formed in the fetus by about 18 weeks and the smell system by about 15 weeks, although it may be 6 months before the nasal passage is free to inhale, Brown. 2008. Although it is somewhat difficult to distinguish between these senses in the watery world of the fetus, since substances can enter both nose and mouth. Ultrasound images, however, clearly show the reaction of the baby to substances introduced into the uterine environment. The fetus becomes familiar with the smell and taste of the amniotic fluid environment, as shown by preference for own mother's amniotic fluid at birth, Brown, 2008. The maternal diet also conveys odors and tastes into the amniotic fluid, which are detected by the fetus as shown by preferential response, head turning etc., at birth, for example, to maternal diet odors such as anise or garlic, Schall et al. 2000, Brown, 2008. The fetus is also more likely to swallow a sweet substance in the amniotic fluid than an unpleasant one, suggesting innate preferences for sweet flavors, Lilly, 1972, Hepper, 1992, and, of more concern, is likely to increase swallowing if alcohol is in the amniotic fluid, Molina et al., 1995. The fetus is clearly immersed in a world of touch, smell and taste. Fetal hearing and auditory memory. The auditory system is also functional to some extent by about 20 weeks, even though the ear and auditory brain regions are not yet mature. The fetus will respond to loud sounds with movements, perhaps showing a startle reflex, Zimmer et al., 1993. By 22 weeks, the fetus will show a more sophisticated reaction by orienting or attending to sounds, as evidenced by movement and a decline in heart rate. Furthermore, if the tone or sound is repeatedly presented, the fetus will eventually show habituation or a decline in responsiveness, evidence of simple learning or memory, Dirks et al., 2009, Leader et al., 1982. The fetus is capable of simple associative learning in regard to the pairing of a sound with a touch stimulus, Hepper and Leader. 1996, but is also able to learn and remember complex sound patterns from the external world. Many natural sounds penetrate the uterine environment, and the fetus can not only hear them but also learns about or remembers them. In particular, newborns show preferential response to the maternal voice, de Casper and Pfeiffer, 1980, and even to theme tunes from television shows watched by the mother during pregnancy, Hepper, 1988, indicating perception and learning of these sounds during fetal life. There is even remarkable evidence of newborn preference for the sound patterns of nursery rhymes, de Casper et al., 1994, or Dr. Seuss stories presented during pregnancy, de Casper and Spence. 1986. More recently there has even been more direct evidence of fetal learning, in that fetuses of 8 to 9 months familiarized to either their mother or a female stranger reading a passage showed a reaction when the opposite voice was then used, Kiselewski et al., 2009. Some of this research, and its role in helping us to understand the development of memory from infancy, is explored further in Chapter 6, Memory in Intelligence. Fetal vision, the least developed of the sensory channels in prenatal life, is vision. The murky light of the uterine world is not optimal for vision, so it is probably just as well that the visual system does not operate fully, or else it may become attuned to poor lighting levels and become ill-equipped to deal with the more intense light outside the uterus. Nonetheless, by about the 4th month, although the retina is still immature, the main parts of the eyes have developed, and the next month sees the specialization of cells along the visual pathways for color and spatial detail. A fetus of 6 months will try to shield its eyes with its hands if a bright light is introduced into the mother's abdomen in diagnostic procedures, Nielsen and Hamburger. 1990, and by the 7th month, the visual region of the brain, the visual cortex, has developed its basic structure with the capacity for analyzing the main features of the visual world, as shown by studies of postmortem tissue, Huttenlocher, 1990. It is thus clear that basic abilities for sensing, perceiving, remembering and learning operate in fetal life, and that the lifelong task of the brain to detect, process and retain information from the external world begins even before birth. Risk to prenatal development, environmental teratogens and genetic errors. Regrettably, not all prenatal development runs smoothly according to the plan outlined so far. It is estimated that about 3% of newborns have some congenital malformation, that is, a problem present at birth, Coulter, 2003. These are due either to noxious or damaging environmental factors known as teratogens, such as maternal disease or environmental pollutants, or to failures in genetic processes. About 10% of congenital problems are due to environmental teratogens and 15 to 25% involve genetic problems, with the causes of the rest being undetermined, Coulter, 2003, Rees and Hobbins, 2006. Environmental teratogens. The embryonic period is the most vulnerable to the effects of teratogens, as this is the time in which all the basic organs and structures including the nervous system and brain are being formed, see above, and so teratogens at this time can be particularly destructive. In contrast, there is little effect in the germinal period. The effects in the fetal period are less severe than in the embryonic period, but nonetheless the sensory organs, especially the eyes, and the central nervous system are still susceptible right across the fetal period up until birth, Moore and Persaud, 2003. Most teratogens either involve maternal conditions or behavior, nutritional insufficiency, stress, ingestion of alcohol, smoking, caffeine intake, etc., or derive from wider environmental factors, such as radiation, Coulter, 2003, Brent and Fawcett, 2007. Malnutrition. One of the most common teratogens is maternal malnutrition. There are many nutrients that are essential to healthy prenatal development, although lack of folic acid, vitamin B9, seems especially serious as it is essential for producing the genetic material, DNA, used in the formation of new cells, including neurons and blood cells. Lack of folic acid is a key factor in neural tube defects, for example, Brent and Fawcett, 2007. Maternal drug use. Legal drugs can be of equal or greater concern than illegal drugs in regard to effects on the developing child. Even the caffeine in a few cups of coffee a day may have harmful effects such as poor growth and high excitability in the infant, Schutze and Zeskind. 1997, and some of the most disabling effects have come from the use of prescribed medications, such as the 1960s sedative thalidomide that produced major deformations of the limbs in the developing embryo, Moore and Prasad, 2003. The most commonly available legal drugs are, of course, alcohol and nicotine, and both of these can have profound effects on the developing brain and body. Ality et al., 2013, Dejin Carlson et al., 1998, Matson et al., 2011. See the case study box. The evidence on the effects of illegal drugs such as cocaine is not as clear-cut because their use may be linked to other risk factors such as poverty and use of other drugs which also have an impact on the developing child. However, maternal use of illegal drugs such as heroin or cocaine is generally considered to lead to low birth weight and disruption of state in the infant, such as poor sleeping patterns, under- or overexcitability. Friedman and Polifka, 1998. Maternal disease. Maternal disease is also another risk factor in prenatal development. There are a number of teratogenic viruses. Rubella is especially destructive in the first few months of prenatal life, when it can cause damage to the developing eyes and ears, heart and brain, resulting in visual and auditory impairments and heart and brain abnormalities. Moore and Prasad, 2003, Rees and Hobbins, 2006. Maternal psychological state. It is not only maternal physical state that can affect prenatal development. Maternal psychological or emotional state also appears to have an impact. Maternal stress or anxiety produces high levels of cortisol linked to growth problems and postnatal cognitive problems. Bergman et al., 2007. For example, extreme maternal anxiety in pregnancy is linked to brain activity reflecting poor cognitive control in the offspring in adolescence. Menace et al., 2009. Environmental toxins, pollutants and toxins in the natural environment may also impact on the prenatal infant, although the evidence in this regard is sometimes controversial. Coulter, 2003. One of the confirmed cases of environmental teratogens occurred in Minamata in Japan in the 1950s, when industrial mercury waste was released into the food chain and water supply, causing prenatal brain damage that resulted in physical and cognitive impairments. Clarkson et al., 2003. There are many environmental teratogens but, as noted above, a greater percentage of problems in prenatal development are due to the effects of faulty genetic processes. Genetic factors that impair prenatal development, as explained in How to Grow a Baby, genes influence prenatal cell growth and development, but there can be errors in this process. For example, a mutation in the sonic hedgehog gene that controls the left-right organization of the neural pathways can lead to the development of a single eye in the center of the face. Gilbert, 2000. One of the most common genetic errors, however, leads to the condition known as Down or Down syndrome or trisomy 21, Tochi, 2000. This occurs for about 1 in every 800 births, with risk increasing markedly with maternal age or most probably the age of the maternal ova which are formed in infancy, the risk rising from 1 in 1900 at 20 years to 1 in 30 at 45 years. Halliday et al., 1995. Most cases are due to the failure of the 21st pair of chromosomes to separate properly during cell replication, usually in the copying of the ovum, so that the new cells have 3 chromosomes for this 21st pair instead of the usual 2. This causes abnormalities in both body and brain, with impairments in cognition, speech and motor capacities, though less severe forms from only a partial failure of chromosomal separation are associated with less marked impairments. Although this list of potential hazards seems daunting, by and large most newborns are not affected by them and make the journey from zygote to neonate intact.